Hello, welcome back. We are continuing our journey through chapter 3. This next little mini lesson is covering cell wall structure. Particularly, we're going to spend most of the time talking about bacteria cell walls. I'll mention a little bit about some differences between bacteria and archaea, which we haven't really talked about archaea yet, but they're a different group of prokaryotes. But when we do get into chapter 4, no, we will talk about archaea in chapter 11. But I have a slide at the end of this chapter that kind of shows all of the differences between bacteria and archaea. So I'm kind of holding off all of those comparisons until the end. All right, so let's take a look at a bacterial cell wall. So first, before we kind of compare the difference between a gram-positive and gram-negative, we have to take a look at a key structural component of bacterial cell walls. It is a large carbohydrate called peptidoglycan. It's a polymer, so you remember polymers, many components hooked together, or polysaccharide if you wanted to go so far as to call it that. But we have kind of the word peptido, peptido means like peptide bond, right? So there's got to be some kind of protein component to it. And glycan, just like we had a glycocalyx, glycan is in reference to sugars. So if we take a look at the breakdown of what this looks like, this is a zoom way in, kind of a diagram of this peptidoglycan. We have two carbohydrates. They're calling it the sugar backbone. We have NAM and NAG, right? NAM and NAG. They stand for N-acetylglucosamine and N-acetylmuramic acid. You don't need to know those names. NAM and NAG is just fine. But if you take a look, they are alternating NAM, NAG, NAM, NAG, NAM, NAG, just alternating. So this is what we call the sugar backbone. That's the glycan part. And then the peptido part is we have a couple short amino acid chains that kind of help hold all of those alternating NAM and NAG subunits together. So we have these tetrapeptide cross bridges. Those are kind of these little green bead looking things. And then we have some connecting amino acids holding those tetrapeptide cross bridges together. So there's connections vertically and connections horizontally made up of these short amino acid chains holding these long polymers of NAM and NAG subunits together in this large molecule called peptidoglycan. What's unique about peptidoglycan, it really is only found in bacterial cell walls. It is not found in any eukaryotic cells. It is not found in any fungal or plant or protist. It's just only found in bacteria. And it is not found in archaea, which is a completely different group of prokaryotic cells. And that was one of their defining features is, hey, here's some prokaryotic cells that do not have peptidoglycan, which we know bacterial cells do. So that's kind of cool. So the major difference between when we take a look at bacterial cells, we can usually categorize them into one of those two groups we saw in chapter 1, gram-positive and gram-negative, thanks to the staining technique developed by Christian Gram. So his name is like forever in microbiology because of that discovery. So let's take a look at a close-up of what a cell wall looks like in a gram-positive organism. So all living cells, they have to have a plasma membrane. And so that's what we're starting with right here. So this is just your regular old phospholipid bilayer. It's going to have integral and peripheral proteins, all of the same stuff you learned back in your general bio. The cell wall sits just outside of that. Okay, and in a gram-positive bacteria, it is a big, thick layer of peptidoglycan. That's going to be different to the gram-negatives that we'll see next. To hold that big, thick layer of peptidoglycan in the cell wall to the cell itself, we have these large proteins called lipoteichoic acids. And that's this big green thing here. And you'll see it travels all the way through the thick peptidoglycan, and then it anchors itself into the hydrophobic region of your plasma membrane. And then we have what are just called teichoic acids. These guys, they don't go all the way down into the lipid bilayer. They just help to kind of stabilize and give some structural support to that peptidoglycan. Now, looking at this, the way that they've drawn it, I mean, it's a pretty good drawing, but it almost makes it look like that peptidoglycan is completely solid, like it's a brick wall, there's no penetrating through. But that can't be the case. Because if it was, the cell would starve. It would not be able to get anything into it or get rid of its waste products. It would poison itself to death. So you have to remember when you're looking at a picture like this, that diffusion will happen in both directions through that loosely organized peptidoglycan. So I can't go back in this when I'm recording on PowerPoint, but flip back to that picture of your NAM and NAG subunits. I'm drawing them very poorly, right? There's gaps, right? So stuff can get through. It's not a solid sealed structure. It is strong. It's the structural integrity of the cell helps to give it its shape, but it has the ability for things to move in and out of it and then be regulated by what the plasma membrane can do. Because we know plasma membranes, phospholipid bilayers are selectively permeable. That all holds true. But I just didn't want you to think that a peptidoglycan is a complete sealed off wall, because this picture kind of makes it look like that. And then I included down here in this picture a light micrograph, and you will be doing gram staining in the lab kit activities. So the purple cells that we can see here, and we've got a couple over here, all of these are the gram positive cells. So this is what they look like under the microscope. Purple is the gram positive, and the pink will be the gram negative. When we get to the staining, I'll talk about why each responds differently to the actual steps of gram staining. But for right now, we're just learning purple is gram positive, big thick layer peptidoglycan. Okay, so here now we are going to take a look at the gram negative structure of the cell wall. So we can start by taking a look at, yes, there's a plasma membrane, right? And it's the same old plasma membrane like we've seen in all other cells, phospholipid bilayer, selectively permeable, hydrophilic, hydrophobic regions, same that we saw in the gram positive cell. The peptidoglycan in a gram negative cell is a lot thinner. So here's our peptidoglycan. It's just a really thin little layer. It's made up of the same NAM and NAG subunits and the amino acid and the tetrapeptide cross bridges, but it's just thin. But what does a gram negative cell have that the gram positive doesn't is this outer membrane. So the outer membrane is, it looks like another phospholipid bilayer, but it's not quite. We can see there's phospholipids on the bottom, right? So they're kind of got the blue phosphate head, and the little yellow lipid tails. But the outermost layer is not. We can see it's pink. So they did it on purpose. It's a different outer structure, hydrophilic structure. It is what we call lipid A. And it has little tails that are kind of hanging down. Let me get a different color. So kind of hanging down this way. And the lipid A is on the top. So it's similar to a phospholipid bilayer, but it's not exactly the same. It can be impermeable to hydrophilic things. So for that reason, there are porins, right? So porins are going to be allowing things into and out of the cell. We can kind of see a cross section of one there. So they're not selectively permeable like a transport channel would be in the normal plasma membrane, the inner plasma membrane. They're just, it's going to be size. If it's small enough to fit through that porin, it's going to be able to move through, because we don't want the cell to be sealed off. This outer membrane would definitely, it's a lot more selective than the open framework of the peptidoglycan. So we need to add some porins in there to allow the cell to get rid of things and bring stuff in. But this outer part, it's called the lipopolysaccharide layer, or LPS layer. That lipid A is actually an endotoxin. We're going to talk about endotoxins a little bit more in chapter 14 when we get to epidemiology and pathogenesis, what can cause diseases. But for now, since we're talking about the gram-negative cell walls, if you have a gram-negative bacteria in you and it dies and its cell structure breaks down, it releases that lipid A into the body, some bad things can happen. So you have widespread vasodilation and increased permeability. You can get a rapid drop of blood pressure, which could lead to shock. There could be fever and vomiting and nausea and a whole bunch of bad side effects. I don't think it's side effects, bad effects of endotoxin poisoning, the release of that broken down outer membrane of gram-negative cell walls. Okay, so that's unique to gram-negative bacteria. Gram-positives aren't going to do any of that. So in our micrograph down here, all of the pink staining organisms are going to be the ones with this structure in their cell wall. Okay. And then lastly, before we wrap up, there's a small group of bacteria called mycoplasmas. You can see this in the note outline if you're following along. Mycoplasmas are a group of bacteria that do not contain any peptidoglycan at all, and they don't contain any cell walls at all. They are cell wall-less bacteria. They are really small, and they were first thought to be viruses, which are infectious agents that lack cell structure. But upon further investigation and better technology, they were able to analyze the mycoplasmas and say, oh, they have plasma membranes and cellular components, more like a cell, not a virus, but they just don't have peptidoglycan, so they get to fit into their own little category. And then lastly, like I said earlier, at the very end of chapter 3, I'm going to compare all of the differences between bacteria prokaryotes and archaea prokaryotes. And this is one of the big differences is archaea do not contain peptidoglycan in their cell walls. They have cell walls, they just don't contain peptidoglycan, right? So we will hit that at the end. Next, we're going to journey to just outside of the cell with discussions on the plasma membrane. Okay, see you next time.